The asthma platform is a mechanistic mathematical model of human asthma that enables evaluation of the efficacy of therapeutic strategies to treat and manage asthma. The platform includes relevant biology related to the pathophysiology of asthma, including airway tissue and cellular life cycle dynamics, inflammatory cell trafficking, and the regulation of these processes by the integrated effect of signals from more than 50 cytokines and chemokines. The local airway inflammatory milieu drives acute and chronic changes in airway biomechanical properties, including airway smooth muscle constriction, cellular hyperplasia, edema, and long-term lung and airway remodeling, which together combine to cause changes in pulmonary function. The asthma platform is capable of simulating the effect of interventions on key clinical endpoints, including changes in forced expiratory volume in 1 second, FEV1, and exacerbations. Additionally, the platform also enables assessment of the relative contribution of reversible changes in lung function associated with smooth muscle cell constriction and irreversible changes associated with chronic lung tissue remodeling to individual patient response to clinical protocols of interest, including antigen challenges. Virtual patients that embody different pathophysiologies of asthma, including obstruction versus constriction dominant and eosinophilic versus neutrophilic, are available for biosimulation research. Human asthma is a heterogeneous disease. The contribution of the different cellular players and their acute and chronic effects on lung inflammation and tissue remodeling likely exhibits considerable interpatient variability, which in turn leads to differences in the severity of clinical presentation as varied efficacy of available and novel therapeutics. The asthma platform contains a diverse set of virtual patients that exhibit variability in baseline disease severity, quality of inflammation, lung tissue biomechanics, and responses to approved therapies consistent with available clinical data characterizing asthma patients. Both acute and chronic asthma therapies with diverse mechanisms of action are represented in the asthma platform, including short and long-acting beta agonists that act primarily to reverse airway constriction, to anti-inflammatory agents that aim to reduce chronic airway inflammation, including standards of care like steroids and montelukast, and more novel interventions like PDE4 inhibitors. Like all PhysioLab platforms, the asthma platform is rigorously validated by comparison of simulation results with published human clinical data, both for baseline measures and in response to interventions. For example, in this first figure on the left, the error bars show the range of reported clinical variability in airway cell populations determined from sputum samples of mild asthmatic human subjects at baseline. For comparison, the cell counts in a representative mild asthmatic virtual patient are shown. The figure shows that the virtual patient exhibits inflammatory cell counts within the realm of feasible clinical variability in human subjects. Similarly, in the second figure on the right, a comparison of clinical data and a single virtual patient response for changes in airway inflammatory cell number following an antigen challenge is shown. Finally, the last figure shows the comparison of acute changes in FEV1 for several mild asthmatic virtual patients following an antigen challenge. These figures illustrate that the virtual patients exhibit responses across multiple measures, both at baseline and in response to standard clinical protocols that are consistent with reported variability. It is important to note that many hundreds of virtual patients representing human patients with asthma of differing severities can be created to span the breadth of reported subclinical and clinical variability across these or other relevant measures. In summary, the ability of the platform to reproduce these clinical phenomena lends confidence to the mechanistic representation of the relevant biology in the asthma platform and to prospective simulations of novel therapeutics and/or combination strategies. Like all PhysioLab platforms, the asthma platform is well positioned to support decision-making across the drug development pipeline.